Welcome back to the third and final part of our osmosis and diffusion lab. Today we're going to record our final observations and the final masses of our eggs. Be sure to record your observations of each of the eggs in our lab. You'll notice that the corn syrup egg here has shrunk, whereas the distilled water egg here has grown. Let's measure this quantitatively using the same methods we've used in the last part of our lab. I want to remove the liquid from our corn syrup egg. I'm going to tare the container, and then I'm going to return the egg to its original container. We see that the final mass is now 48 grams, quite a bit less than it was initially. I'm going to do the same exact procedure with the distilled water egg. I just remove the liquid, place the container on the scale, tare or zero the container, and return the egg to that container. In this case, we have a mass of 98 grams. That is quite a bit more than it was initially. That concludes our lab. Be sure to record your data and observations and move on to your analysis. In our lab, we found that the egg in corn syrup lost mass or shrunk. This indicates that water moved out of the cell and into the surrounding environment. This egg was in a hypertonic solution. Remember, cells in a hypertonic solution have solute concentrations lower than the solution that bathes them. Another example of a hypertonic solution would be a cell in salt water. The egg in the distilled water grew, indicating that it absorbed water. This is because it is in a hypotonic solution. Remember that cells in hypotonic solutions have solute concentrations greater than the solution that bathes them. This concentration difference causes water to move from the surrounding solution into the cell. A simple way to remember which way the water will travel in osmosis is the phrase salt sucks. But it applies to more than just salt. It can apply to just about any solute. If there is more salt or solutes in the environment, the water will be drawn out of the cell. If there are more salt or solutes inside the cell, the water will be drawn into the cell. This concludes our series. I hope this lab has helped you understand the process of osmosis.